is the second undergrad we have working with us. His name is Sam Cranston. He is a senior biology major with a double minor in chemistry and history. Sam is from Martha's Vineyard, and if you've ever been there, you know there's a lot of ticks. And so he started his research on ticks back in high school, where he worked on a project aimed at slowing the spread of Lyme disease on the island. Sam was very eager to join the lab here, and he started working in his junior year as a lab assistant to Alyssa Newhouse on her malaria project. And then this year, Sam spent his project working on ticks, which is he's going to present on, and he's kind of got our lab super interested in ticks, which is really nice. And I really am excited to announce that Sam just accepted a job as a research associate at Boston University's Center for Regenerative Medicine, where he'll be studying novel stem cell treatments for blood-borne diseases like sickle cell. So it's a huge
Thank you, Erica, for that introduction. I'm Sam Cranston. I'm going to be presenting on my research on black-legged tick infection dynamics in northern Vermont forests. And thank you all for being here. So first, again, as Matt started, I just want to acknowledge my amazing mentor, Dr. Ellen Martinson. None of this research would have been possible without her help, and I'm truly, truly grateful to her for all her advice and collaboration on this project. I'd also like to thank the UVM Biology Department for me receiving the Barbara K. 67 Award for Summer Research this summer, which allowed me to do this project. It wouldn't have been possible without that, or funding from the University of Vermont 4 and the University of Vermont Career Center, as well as all the members of the Wildlife Pathogens Lab. So thank you to all those people. You'll hear more about them at the end. So first, I just want to start with some background about myself. Like Erica said, I'm from Martha's Vineyard, Massachusetts. So as scientists, we know that island ecosystems behave differently than mainland ecosystems, particularly when it comes to biodiversity levels. So Martha's Vineyard is no different. It harbors really high populations of white-footed mice and white-tailed deer, which, if you know anything about the ecology of tick-borne disease or tick-borne pathogens, that's a really big problem. And so I have these stats up here, but the main one I want to highlight is 10 to 30 times greater disease rates found on Martha's Vineyard than on mainland Massachusetts. So I live in Vineyard Haven, and it's a 5-mile difference across that Vineyard Sound, and there's 30 times greater tick-borne disease in humans on Martha's Vineyard. So it hits close to home for me. I know so many people who've had their lives impacted by these diseases. My best friend in high school missed all of her senior year because she had chronic Lyme disease. Teachers have had chronic Lyme disease, and the list goes on. My family members, my dog. So it's a big issue, and it got me interested in this. And as I'll show, it's not just a problem on Martha's Vineyard, but a big problem here in Vermont as well. So yeah, this is just showing how black-legged ticks have expanded in recent years, mostly due to global warming, allowing them to survive in habitats they couldn't have previously survived in. So this is just showing the cases, human cases of Lyme disease per year, and you can see how rapidly it's expanding northward, and also I want to highlight the state of Vermont. So we're situated in a perfect place to study these pathogens, as we're right on the edge of this wave of tick-borne disease expansion. And so I decided to continue my studies here and research these pathogens within Vermont's black-legged tick populations. So again, just some more data showing that this expansion is indeed happening here in Vermont, and just like some alarming facts. Over the last 10 years, Vermont has the highest increase of Lyme disease out of any state in the US. And between 2010 and 2019, reported cases of Lyme disease rose by 744%. So we're clearly experiencing a big expansion of these black-legged ticks, which are also, black-legged ticks and deer ticks are interchangeable terms, just so you know. So we're experiencing this big range expansion, and their associated pathogens are coming with them. So this map is a sampling effort done by the state of Vermont. So green towns are where they sampled, and they only collected 23 black-legged tick nymphs, which I'll get into more, but that's the most relevant stage to human disease, or the most relevant life stage to human disease. So that's all to say that there's been a limited sampling effort by the state of Vermont in recent years for reasons that I don't need to get into here, but hopefully we see changes in that in the future with more studies such as this one coming out of UVM and similar institutions. So I just wanted to go into the cycle of tick-borne disease and kind of explain why the nymph is the life stage that I chose to focus on in this study and why it's the most relevant to human disease. So this graph is showing the Vermont tick-borne disease report from 2018 and the months where we saw the highest incidences of Lyme disease in particular. So you'll see that it's June, July, and August are the main months where we're seeing a lot of tick-borne disease cases. And if you look at this map from this, or this cycle from the CDC, that's when the black-legged tick nymphs are active. And so adult ticks carry higher levels of pathogens, but nymphs are just so much smaller, as you can see from this size chart. So they're much more easily missed on tick checks. So I actually have some tick samples. If you guys would like to look at them. Sorry, Matt. So we have, there's an adult male, adult female, and a nymph. So it will really, like, this doesn't even do justice. I think that nymph is too big on that. That doesn't do justice to size comparison. So it's really important that we're looking at nymphs in particular because they're just, like I said, more relevant to human disease. And then I just wanted to highlight this chart. So like, you can see this cycle, but this is really, this is tick-borne disease or tick-borne pathogens and its relation to humans in a nutshell. So essentially, an uninfected tick will bite an infected reservoir host, which in this case is depicted as a white-footed mouse, but could also be a raccoon, a squirrel, a number of other small mammals, and even birds that ticks are biting. And so they're picking up these infections from the reservoir hosts and then become infected themselves and then pass those infections back to uninfected reservoir hosts. So the cycle continues, right? And so if you increase populations of either one, you're gonna increase incidence of tick-borne disease likely. And they're not actively searching for humans, but they will bite humans and pass these pathogens on to you or your loved ones. So it's kind of a misconception that deer play like a central role in this cycle. Deer are definitely involved in it, and you can think of the deer kind of as like a singles bar for the ticks. So like that's where the adult males and adult females are meeting and mating. And then females that survive the winter will lay eggs in the spring and populations will expand. So as we're experiencing these milder winters, more females are overwintering, laying more eggs, and tick populations are increasing exponentially because of it. So when you think tick-borne disease, you think Lyme disease, right? Borrelia burgdorferi. And that's obviously not wrong. Ticks spread a lot of Lyme disease. They also spread a host of other pathogens, such as Anaplasma phagocytophilum, the bacteria that causes anaplasmosis, and Babesia microti, which causes human babesiosis, as well as more pathogens. We're only capable of screening for 3 pathogens in this study, but there's a host of pathogens that these black-legged ticks are carrying and transmitting to humans. So that brings me to my central research question. How are the pathogens Borrelia burgdorferi, Babesia microti, and Anaplasma phagocytophilum distributed in populations of nymphal black-legged ticks in Chittenden County, Vermont? So these were our methods in order to answer that question. So as I showed in that previous graph, ticks, nymphal black-legged ticks are really only active during June, July, and August. Like not really even into August. We stopped finding them at that time. So really June and July. So this is how I spent my summer in the fields collecting black-legged ticks. We use these drag cloths. So you drag them along the ground. The ticks think it's a small mammal or something that they can bite onto and take a blood meal from. They bite onto the cloth and we take them off and collect them. So that's me with my first nymph that I collected. So it's clearly really, really happy about that. And that's Ellen, Dr. Martinson, and Lauren Berkeley in my lab. And also this is Lena, Ellen's daughter, and Hannah were really helpful in my project. They're actually here today. So they did a lot of help with me collecting ticks this summer. So big shout out to them. And this was a real learning experience for me and my career as a scientist, hopefully, because it seems like a straightforward process, right? Like field collection, DNA extraction, multiplex PCR, gel electrophoresis. But as scientists, we know that no science is just straightforward like that, and we hit a lot of roadblocks along the way. Ellen, Dr. Martinson, and I had never field collected before, so it took us a while to perfect our field collection techniques. And then developing a multiplex PCR protocol is a lot easier said than done. And so it took a number of weeks of troubleshooting for us to finally be able to visualize all 3 pathogens on a single gel. But we were able to do that, which allowed us to visualize co-infections of these pathogens, which is what we're really interested in this study. So these were my field collection sites. We were initially looking for differences on a site-by-site basis, but we didn't find any significant results in terms of pathogens levels throughout these 5 sites. So that led us to kind of pooling our pathogens, or all our samples together. But I just would like to highlight Centennial Woods, which is, I'm sure as many of you know, right there. We didn't do density estimates because that's really hard to measure, but from my own observations, that was by far the highest density of black-legged ticks we found anywhere. Yeah, so that's concerning because as professors and students of UVM, you know that so many students go there for labs or just hang out there. And I've been there before for many labs, and I did not know that there was that many ticks present there. But it was frankly a really concerning amount of ticks, and people go there every day, so it's important that people are aware. And that's not to say that there wasn't high densities at these other sites, there was, but just we were kind of astounded at the amount of ticks that we found in Centennial Woods. So that brings me to the results of this study. So like I said, we pooled our data across all 5 sites. And what we found, this is the Vermont tick pathogen surveillance report from 2019, so that's the most recently available data they have on NIMS, and this is the results from this study. So as you can see, we found higher prevalence of all 3 pathogens than the state found in 2019, by significant margins for Borrelia and Anaplasma and slightly more Babesia. So there's a number, we've been thinking why maybe we saw this. There's a number of reasons why, and one could simply be that these pathogens have expanded over these past 3 years. I just mentioned how we've seen a 744% increase in Lyme disease cases over 10 years, so it's not too far of a stretch to say that in 3 years these pathogens have simply extended further in Vermont black-legged tick populations. And also, the state is looking at data on a statewide basis, right? So we only collected ticks from Chittenden County, and so it could just be that Chittenden County has higher prevalence of these pathogens in their tick populations. And that makes sense because tick numbers decrease as you go up with elevation, and this is the most highly urbanized area of Vermont, so that could be impacting the levels of pathogens that we're finding. And just the last thing I want to highlight is the state doesn't do any testing for co-infections of these pathogens. So we were really interested in co-infections of these pathogens and found significant levels of co-infections. We didn't expect to find any triple infections within our pathogens, and we found a 1% prevalence of that, which is not insignificant and quite frankly a lot more than we expected to find, and it's not being looked at, like I said, in Vermont's tick populations. So this is a permutation analysis of the infections we saw. So it's representing deviations from statistical expectations of infections and basically showing how these pathogens are non-randomly sorted within the tick populations. So if this graph was 0, if these bars weren't present, the pathogens would be completely randomly distributed throughout black-legged tick populations. However, that's clearly not what we found. We found a heavy negative correlation away from these pathogens existing as single infections and a bias towards these pathogens associating with one another and existing as co-infections. So just for example, I wanted to point you guys to this chart on the right. The observed frequency of anaplasmosis as a single infection, so just an anaplasmosis infection, was 3.35%. But if you go back to this graph, we saw a 10% overall infections with anaplasmosis. So what's that? That's saying that 65% of the time that we saw, greater than 65% of the time that we saw anaplasmosis infection, it was together with either Borrelia or Babesia, or both of them as a triple infection. So that's concerning, one, but also as disease ecologists, that let us question, why are we seeing this? Like, why is there this really heavy bias towards co-infection in the black-legged tick nymphs that hasn't really been studied across the country, but in Vermont in particular, and there's just not a lot of data on why these co-infections are happening, and so that's why this study was cool. So it's obviously a complicated system, right? And we can't exactly answer that question right away, but when thinking about black-legged tick nymphs, they've only taken one blood meal. So they've only had one chance to pick up these pathogens from a reservoir host. And so that means that whatever reservoir host they bit initially on their first blood meal was carrying a co-infection likely. And so from a 2014 study from Hirsch et al., that's a really good study, they were able to show that all the organisms I've depicted here, as well as others, are competent reservoirs for all three pathogens that I've been interested in. So that means that they are able to harbor these pathogens and are reservoir hosts for these pathogens. But the levels at which they're carrying them are extremely variable. So like a robin, for example, less than 1% of robins may be carrying any of the three pathogens, Borrelia, Anaplasma, or Babesia. But the white-footed mouse and the eastern chipmunk are incredibly competent reservoir hosts, and they're carrying really high levels of these pathogens. And so the fact we found so many co-infections indicates that these ticks are likely feeding on white-footed mouse and eastern chipmunks and are less likely to be feeding on these other reservoir hosts. So like, ideally, we'd love it if all of our ticks are feeding on possums, right? Because possums have really low infection, and they bite off and kill a lot of the ticks and don't even let the ticks bite them in general. But from my data, it's pretty clear that that's not what we're seeing in Vermont. And that's not necessarily, that's a kind of concerning trend. And so as we all know, we've been seeing a strong decrease in biodiversity with global warming and climate change and just human impacts on the environment. And so this is taken from Chittenden County. As we continue to disturb our environment, these species are really resilient species and have large, strong populations. Like you could go outside and probably see 3 or 4 chipmunks just walking across UVM campus. But you're not necessarily going to see a possum or rare ground nesting birds that ticks also feed on. So as we continue to lose biodiversity and continue to impact our environment, ticks are going to continually feed more and more exclusively on these 2 really competent reservoir hosts. And the problem of tick-borne disease in Vermont and across New England is just going to continually get worse. So again, just wrapping it all up, why is this important? We showed that co-infections are relatively common in Vermont's black-legged tick populations. Studies in co-infections are important because they can provide a better understanding of how these pathogens are distributed throughout the environment, how they may be associated or not with one another. And off that, land use changes have had significant impacts on biodiversity at all levels of the ecosystem, so down from the top predators to the pathogens that are all part of the ecosystem. And this is all to say that education is the best solution to this problem, and increasing our surveillance, just increasing our understanding of how these pathogens are distributed, and just preventing yourselves from getting bitten by ticks in general is really the best solution to staying safe from this problem. So understanding how dangerous they truly are and being aware that these biases towards co-infections do exist and just preventing them from biting you in the first place is the best way to not get tick-borne disease. So again, I really want to thank my research advisor, Dr. Ellen Martinson. She's been amazing throughout this project, and I truly would not be where I am today without her amazing advice, and just her passion for advising all of us in the lab really comes through. And thank you, Ellen. And thank you to the members of the Wildlife Passions Lab, Erica, Lauren, and Matt. I would come in every day with like the most random questions about disease ecology, and they're all so equipped to answer those questions and been just amazing partners in this experience and really like a little family that we have up here. So I'm really gonna miss you guys next year. And I want to thank a couple other people. My high school mentor helped me throughout this project. Tucker Perrin, who's not here, did a lot of help with the data analysis. So thank you, Tucker, wherever you are. And my parents. Thanks, Dad. Thanks, Mom.